Okay, good morning, everyone. Thanks for being here. So I'm gonna start this talk, which is devoted to these two software tools. One is called MEIGO, the other one is a related tool called saCeSS. And first of all, I will start by providing a bit of motivation about the reason we developed these tools. So these days, we have all this omics data coming from new technologies that are improving all the time. And we need to make sense of all this data. We need to be able to distill or extract some insights, some meaning, some knowledge from all these omics data. This is, of course, a very general problem. Very roughly speaking, we can say that there are two types of approaches to deal with that problem. One would be the typical data-driven approach, which is, of course, being used in statistics for many, many years, but in more recent years, it's the topic target of machine learning and many other artificial intelligence methods. So basically, in these data-driven approaches, we just need the data, and then we have some methods, some data-driven model methods, and we can perform different tasks like classification, regression, and so on. But then there is a second approach, roughly speaking again, that is more based in first principles or previous knowledge. Sometimes it's called knowledge-based approach, prior data or prior knowledge approach. And here, not only we depart from data, but we also depart from prior knowledge that we have about the biological system under study. And this prior knowledge is usually encapsulated, is usually provided as a model, usually a mechanistic model. Okay, so we have some theory, we have some evidence of some mechanisms going on, and we encapsulate that knowledge into a mechanistic model. For example, in our groups, like in many other groups, we use differential equation models, but this is not the only way. But these mechanistic models are just a way to encapsulate, to add this prior knowledge to our analysis. And with these methods, we are able to do a series of predictions with simulations and many other tasks. So this mechanistic model approach or knowledge-based approach is very typical of what has been called systems biology. It's not the only approach in systems biology, but is one of the main approaches. And in fact, in this review paper that we published some time ago about reverse engineering, meaning reverse engineering means recover mechanistic models from data, okay? In this review, we were exploring why these problems are so difficult, why these problems are hard in general, and what are the different perspectives from different areas to attack this problem of reverse engineering, recovering this mechanistic model from data. And one of the components is, of course, optimization. And the two software tools that I will describe today are, by the end of the day, optimization toolboxes devoted to problems in systems biology. So the view here is that we can use mathematical optimization, and I will describe in the next slides what I mean by that. We can use mathematical optimization to build models from data, then using those models to design, for example, optimal intervention. An example would be in metabolic engineering, you want to find out which knockouts you should do to improve the production of a metabolite of interest, for example. Or it could be if you are trying to locate a drug target to, to cure some disease, you know that this disease is the consequence of some malfunction in, for example, cell signaling pathway that would be typical of cancer, for example. and you want to design the best intervention to avoid the pathological state. But we can also use optimization for explanation and understanding of the models that we have previously recovered. Okay, so that would mean why is this pathway as the product of evolution? Why has these regulatory networks and not other possible ones, for example? So trying to understand the way these pathways work, trying to distill the principles that evolution has chosen for these pathways to be the way they are in the present time. And then finally, in this other related field of synthetic biology, we can also use optimization to find the best designs for a new part that we want to insert in a living system. So let's start by defining what is mathematical optimization. The very brief definition is just to find the best solution to a problem from the set of possible ones. And by possible ones, we are meaning those that meet some requirements that we have declared in advance. And these requirements are usually expressed as what is called the constraints, okay? Typical examples from everyday life is finding the shortest distance in a map, okay? Which is trivial in a paper map, not so much in a map on a sphere and so on. Another classical problem in optimization is the optimal diet. You want to, for example, create a diet, a meal plan that minimizes the cost, but meeting at the same times the nutritional requirements. Or you want to minimize the amount of calories while meeting these daily nutritional requirements, like calories, protein, vitamins, and so on. In fact, this optimal diet problem was one of the first solved with modern optimization methods, which were created back in the 50s. And another example, it's you have to commute from your home to work, and you want to find the fastest route, but you need to take into account constraints like speed limits. Maybe you have to drive on the highway for some time, and there is a speed limit there. But for example, one day you find that there's a road or street that is closed, so you need to find an alternative way, but you still want it to be the fastest. So this is an example. You are trying to minimize your time to work. That's your metric for the best solution. But you need to take into account constraints, which again are which roads or streets are open or closed, and what are the speed limits. So this is very general mathematical optimization introduction in 2 minutes. Now, we usually express these problems with these 3 components. We say find the x, where x is a vector, and x encapsulates your decision variables, okay? For example, in the example of finding the meal plan to minimize the cost of your diet subject to constraints, x would be the composition of your meals, okay? And here you want to minimize or maximize something. Minimizing something is equivalent to maximizing something if you just change the sign. So minimization and maximization can be reversed, no problem. And then this must be subject to constraints. And constraints, they can be of 2 types. They can be equality constraints, something that must be met with an equality. And typically, this is the model of your system. And inequalities, something that must be larger than or smaller than some other condition. So optimization is a huge area. It started with something called linear programming in the late 40s, early 50s in the past century. But now it's a very large and very mature area. And there are many, many different kinds of optimization problems. and they differ in the way in the form of the objective functions and the constraints, if they are continuous or discrete, if they are nonlinear or linear, and so on. So for each class of problems, many people have devoted especially tailored solution methods that are very efficient. But still, many of these problems are very, very hard. And one of the examples of very hard problems is global optimization. I will explain what is global optimization in a minute. So some time ago, we published a small review of the uses of mathematical optimization in systems biology. And you can see in this table that going from very simple optimization problems, for example, when the objective function is linear and the constraints are linear, this is called linear programming, okay? We can solve these days linear programming problems with huge sizes without any problem in any small computer. And a classical example of linear programming application in systems biology is what is called flux balance analysis. And I guess many of you are familiar with this FBA term, which is probably the most successful or most popular kind of optimization problem in systems biology. Then in this table, as you go down, you go to nonlinear programming, bi-level optimization, mixed integer linear programming, and so on. Things get more and more complicated, but you can see that for all these types of optimization problems, many people already have already developed specific applications in computational systems biology. So the message here is optimization in systems biology is a useful tool, and it's a tool that's being used by many, many researchers. So coming to the topic of this talk, I'm going to talk about two software packages. The first one is MEIGO, and there is a related package called saCeSS. Okay, the difference is that MEIGO was initially the first toolbox that we developed for global optimization. And you can see here the link where you can download the software and the documentation. And saCeSS was a more recent addition where we rewrote the MEIGO code, part of the MEIGO code, to be able to run it on clusters and supercomputers. So MEIGO, there are actually two toolboxes. One is written in MATLAB, another one is written in R. And MEIGO, in principle, is the tool that you would like to use when your problem can be solved on a laptop or a workstation. However, if your problem is larger, much more difficult, computational costly, then MEIGO is probably not the best approach. And this is why we created saCeSS, which is written in C and using two ways of parallelization, MPI and OpenMP, and saCeSS is intended to be run on supercomputers and clusters. You can also run it on a modern workstations where you can compile C with MPI and OpenMP, but it's intended for especially high-performance computing hardware. So first, I will talk a little bit about MEIGO. So the main paper where MEIGO was described was written back in more than 10 years ago already. And this was a collaboration between our group at CSIC and the group of Julio R, who at that time was at European Bioinformatics Institute near Cambridge in the UK. He's back there now because, well, during the last decade, he was in several places in Germany, more recently in Heidelberg, but now he's back and he's, I believe, the head of research of European Bioinformatics Institute at this moment, since last year. So this is the paper, and you can see the paper has already 11 years. However, the MEIGO toolbox, we have kept improving it and debugging it with the help of the feedback of many users. And the latest version, you can get it from this link. So not from the link of the original publication, unfortunately, but it will redirect you to this other link where you can download the latest version. So essentially, what is MEIGO? Well, it's a toolbox devoted to metaheuristics. I will mention in a minute what I mean by that. Metaheuristics are intended to solve global optimization problems, which, as I mentioned before, are extremely hard. And the toolbox is not general in the sense that it's intended to solve general global optimization problems, but is intended to solve problems that are mainly from the areas of systems biology, computational biology, and bioinformatics. Okay, and essentially, MEIGO has two metaheuristics, enhanced scatter search and a cooperative version, which is meant to be run in parallel machines, and VNS, which is variable neighborhood search, with also its parallel implementation. Again, as I mentioned before, when you want to declare a problem to be solved with this tool, you need to express it in a way that is consistent with the standard optimization problem statement. Just to clarify what I mean by global optimization, imagine that you have a toy problem where you only have two decision variables. So you can make a plot of your function f, which depends on only two things, x and y. And it might be that your function has, in this case, a unique global maxima, okay, maximum. Or if your function is multimodal, there will be many local optima peaks and one global solution. So global optimization seeks to find the global solution to a multimodal problem. Okay, and in general, we call these problems non-convex. So non-convex optimization and global optimization can be considered to be the same thing. This is an area that has been studied for a number of decades already. There are many books, huge amount of papers on the topic, many different methods. As I say, the main takeaway message is that global optimization in general is a very hard problem. So there are many methods to solve it, but as you can suspect, when there are many methods to solve a problem, it's probably because there is no one single method that can solve efficiently any arbitrary global optimization problem. And this is the case. So we have on the one side deterministic methods, which are very nice because they can give you mathematical guarantees, certificates of global optimality, so to speak, that you have found, in fact, the global solution to a problem. And there are many types of deterministic methods. The problem is that they do not scale up well with problem size, meaning if you have a small problem, typically less than 10 decision variables, they work quite well, quite fast. But for example, for nonlinear problems, of course, if you have more or many more, then the computational cost explodes. Just they can still provide you with a global certificate of optimality, but you might need years or centuries of computation. So they are not practical for many real applications. Then we have what some people call stochastic or probabilistic methods, and there are many different families, and you will probably recognize popular methods like simulated annealing or genetic algorithms. So genetic algorithms are usually understood as part of a broader family called evolutionary computation. Well, there are many methods in this category. And they work reasonably well, even for relatively large problems. But the price to pay is that you don't get guarantees of global optimality, okay? So sometimes it's enough if you found a good enough solution for a problem, but you don't have the certificate of global optimality that deterministic methods do have. However, the advantage over deterministic methods is that you can solve many real-life problems with these methods, okay? And then we have hybrids, which try to combine both approaches, okay? Try to take the best of both worlds. And inside these hybrids, there are what people call metaheuristics, which are essentially guided heuristics. So they are still probabilistic in nature in most cases, but they combine stochastic methods with some typically local deterministic methods. And one of the methods that we have found to be particularly good for our problems in computational biology and in system biology is based in what is called scatter search. So scatter search is one of the main components of the MEIGO toolbox. So again, this is the typical definition from Wikipedia of a meta-heuristic. Again, it's just a guided heuristic, okay, that can give you a sufficiently good, usually very, very good solution, but you don't have the guarantees of deterministic methods. This is another classification of meta-heuristics. And you can see that there are many kinds. Again, you can recognize here simulated annealing. You can see here genetic algorithms, and scatter search, you see it here, can be regarded as part of evolutionary algorithms, but with other components which make it quite different from, for example, a typical genetic algorithm. And it has been our experience and that of many others that for the class of problems that we are talking about, scatter search is much more reliable and efficient in terms of speed of convergence that, for example, genetic algorithms or simulated annealing. So again, we have MEIGO as implemented in MATLAB in R, implemented scatter search for continuous problems, and variable neighborhood search for integer programming, where the decision variables are integers only. I will now talk very briefly about what is each one of these. So on the one side, you have scatter search. As I say, we develop it to be applied to continuous nonlinear programming problems or mixed integer nonlinear programming problems. The difference here is that some of your decision variables are integers, some are continuous, and this is very common in what is called parameter estimation in systems biology, okay? And then variable neighborhood search is meant for integer programming, where all your variables of decision are either integers or even binaries. And you can solve problems like structure optimization, feature selection, combinatorial problems in general. For example, finding the knockouts in metabolic engineering is a typical application. We also, by the way, in the R version only, not in the MATLAB one, we also included a Bayesian fitting method because some people also are fond of this type of approaches to be compared with our other metaheuristics. Now, to use this from MATLAB, you need to declare the objective function and the constraints. And of course, you can do this in a very compact form. This is just a toy problem. You just solve here very simple function. But instead of this very simple function, you can call a simulator of a huge model that can be expressed in the form of differential equations or stochastic differential equations or some other form. Could be an agent-based model, can be anything, can be a black box, in fact. You just declare the objective, the constraints, if you have only bounds, or if you have inequality constraints, and then some other settings for the internal solvers, and then you call ESS and you get a solution. For R, the toolbox is very, very similar. It was implemented by essentially the same authors. Again, the PIs at the time were Julio R Rodriguez from EBI and me, and Jose Gea was a former PhD student of mine. There were other members of the two groups here involved, and this was done when Jose here was already a postdoc researcher. And in R, you can see another simple example here. You also have to define, obviously, the same components, the objective function, the constraints, and then you call the solver indicating which one of the meta-heuristic, in this case, enhanced scatter search, ESS, you want to use. Okay, let me give you some real examples from the field of computational biology and systems biology. Typical one is you have a model, for example, a model in ordinary differential equations. You have time series data. You want to calibrate the model to the data, meaning you want to estimate the parameters of the model that give a good fit to the data. This is called the parameter estimation problem. It's a so-called inverse problem. And you want to minimize, your objective function is now to minimize the distance between the data and the predictions of the model. So this would be the mathematical expression. We don't really have to go into very technical details here. But for example, if you take the R version of MEIGO, you can very easily load one of the examples that is devoted to parameter estimation. So you just load in R the MEIGO code, then you load this example. And in this example, there is this model, this logic-based differential equation model. There is some data which was generated from the model itself. So we know that the model should be able to match the data, but we just added some noise. So this is synthetic noisy data, just to check that the method is working. And when you call one of the solvers in MEIGO, in this case, ESS would be the best choice to solve this problem, you will get a very good fit to the data as expected, because the data was generated, in fact, with the model. However, the problem is a global optimization one. If you try to solve this problem with a local solver, you'll probably end up in local solution, which in this case would lead you to think that you cannot explain the data with the model, when in fact you can, because you created the noisy data with the model. So this is a good example of parameter estimation problem. Another example that is also included in the paper, in the MEIGO paper, is an application in metabolic engineering. So imagine that you have a relatively large model of the central carbon metabolism of E. coli, okay? And you want to do some metabolic engineering, meaning you want to carry out some knockouts to increase the production of a metabolite of interest, for example, succinate, okay? So in this case, you can download this E. coli model. In this case, this is a constraint-based model, as typically used, for example, in flux balance analysis. You can download it from the website of Bernard Paulson at the University of California, San Diego. And then we can solve the problem to find the knockouts that give us the best succinate production. In this case, we need to use the discrete optimization method, VNS. And you will see that if you do 10 runs, not all of 10 will give you the best solution, which is this one, which corresponds to 25 units of succinate production. Sometimes you will get 20, sometimes you will get very bad production. Why? Because the problem, this is a discrete optimization problem in nonlinear space, is very hard. But even though VNS cannot guarantee you to give you the global solution, you can see that in 10 runs, in 7 of them, you will get this solution, which is supposed to be the best, okay? If you now compare this with a discrete genetic algorithm, you will see that now in the 10 runs of the genetic algorithm, you only get the best solution 2 times, okay? So you can see the advantage of using the metaheuristic that we provide in method versus using a more or less standard discrete genetic algorithms. The reliability and the efficiency of VNS is much better for this kind of problems. And this was, as it was also the case for scatter search, this was the motivation to include this method and not a genetic algorithm or simulated annealing. Now, of course, in addition to these very small problems, we have, along the years, we have done a number of benchmarking exercises. One of the most recent one was done with a group from University of Bonn in Germany. And with our colleagues, we applied scatter search and other supposedly state-of-the-art methods to the calibration of large-scale dynamic models of systems biology processes. And considering all the benchmarking, again, scatter search as implemented in MEIGO was the best performing approach. We can also see that for other problems, when they are much larger, then we need to move to this parallel optimization library saCeSS, which was created in 2017. So now I will talk about this other toolbox. So saCeSS means self-adaptive, cooperative, enhanced scatter search. So essentially, it's an implementation of scatter search for high-performance computing machines. And why is self-adaptive and cooperative? Because the method itself cannot be parallelized easily, but what we can do is we can run in parallel several instances, and they can cooperate among them. So it's like an agent-based optimization approach. So this was the paper which appeared in 2017. Since that paper, we have improved the method and the code in a number of ways, and you can download it from this Bitbucket link. And essentially, when used with large problems, we can get, for example, here you can get even superlinear speedups, even 2 orders of magnitude, just we using 10 processors. So the parallelization and this cooperative approach did end up in very, very large speedups for many problems. And essentially, to declare a problem in saCeSS, now you need to, unfortunately, write more low-level code, but you can actually define the problem using XML. But you need to be not a good programmer, but at least you need to know your way in C and also in compiling in supercomputers or clusters and how to properly use OpenMP and MPI. So here it's not as usable. You need to be more knowledgeable about high-performance computing, but of course, for large problems, is the only tool that we recommend. In a more recent benchmarking exercise, again with our colleagues from Germany, now we are comparing saCeSS with other parallel optimization methods. And you can see that we compared it with other 35 solvers on 28 very difficult problems using 2 different supercomputing infrastructures. And again, saCeSS and some saCeSS variants here in the top were the best performers. In a recent collaboration with people from the UK, from Oxford, we also applied this to a problem that they were having a lot of trouble to calibrate. This was the calibration of a model of cell cycle. And in fact, saCeSS was the only method able to calibrate it. And this is detailing a publication that appeared last year. We also have another recent collaboration with people from Marseille on the calibration of a whole brain network model of, in this case, devoted to study epilepsy. Then again, we compare our method saCeSS with other parallel optimizers, and ours was clearly the best performing. So takeaway message, optimization is key. Also in computational biology and bioinformatics, you can solve many important problems with it. Many of these problems are non-convex. This means that you need global optimization, and these problems are very hard. And in many instances, you even have mixed integer problems or black boxes. So there is this need of global optimization methods, and this is why we have developed these 2 toolboxes. I include here the links, but all these links are available in our website in the section devoted to software. And with this, I end up my talk, and I can now, of course, take questions.